Okay, well, it looks like we're just about all here, and I'm going to go ahead and get us started. Anybody have any questions on anything before we start? Okay, if you have any questions, send me an email or throw it into chat, and I'll try to answer it right away, or I'll have to say, if I have to think about it long, I'll send you an email. But otherwise, we're going to go ahead and get started here. Now, today we're going to wrap up chapter 3 on the cells. We are going to start here on protein synthesis. I hope everybody can see the screen. It says protein synthesis. Everything, the glitches earlier in the week seem to have stopped. So hopefully that's the way it stays. Now, we talked about protein synthesis a little bit in the sense that the ribosomes are where the proteins get made. The ribosomes are located on the rough endoplasmic reticulum. They are made of ribosomal RNA. Now, all the RNA in a cell gets made in the nucleolus inside the nucleus. But how do we get proteins from DNA? Well, it has to do with the order of the bases. The code to make any protein is determined by the specific order of the bases. Now, there's only 4 bases: adenine, guanine, thymine, and cytosine. In any, throughout the length of the DNA strand, you have, these 4 bases in any, it appears to be a random order, but it's not random. It's a very specific order. The order of the bases determines the order of the amino acids assembled into a protein. And the order of the amino acids is very, very specific. And I've given this example before, and you're going to get tired of hearing it. But hemoglobin is made up of 360 or so amino acids in a very specific order. The 6th amino acid in hemoglobin is glutamic acid. It has to be glutamic acid. There's only 20 amino acids. I know we repeat a lot since there's only 20 there, but the number 6 amino acid in hemoglobin has to be glutamic acid. If by chance it's valine, it's not by chance, it's by genetic dictates. If the 6th amino acid is not glutamic acid and is instead valine, then the hemoglobin is completely messed up. That's a technical term, messed up. It reduces its ability to carry oxygen by about 80%. It causes the hemoglobin molecule to get very brittle and crescent-shaped, and it causes the red cell to become crescent-shaped, and we have what's known as sickle cell disease, because the red cells take on a sickle shape, all because of one little mistake early on in the production of the amino acid, production of the protein. The wrong amino acid is put into the number 6 spot, and that becomes a significant mistake. It's one little substitution, but it becomes a very significant mistake. And that doesn't happen randomly. It happens because sickle cell disease is a genetic disorder that has to be inherited. It isn't something that you acquire. So the code for the order of these amino acids in a protein, every protein is made of amino acids. The order of the amino acids is dictated by the order of the bases in the DNA. The DNA is the code book, the source book, the information manual, if you will, a cookbook. It contains the instructions on how to make every protein. And that's what we are. We're proteins. we're made up of proteins and minerals and ions and stuff like that. But it's proteins that we use to put everything together. And the proteins are assembled in a specific order, amino acid by amino acid. And the order of the amino acids is represented by the order of the bases in the DNA. So the DNA determines what proteins are going to get made. Now, to make it a little simpler, the bases are organized in clusters of 3. They're called a triplet. So we have a triplet of 3. Adenine, thymine, guanine is a triplet. Cytosine, cytosine, cytosine is a triplet. Guanine, cytosine, guanine is a triplet. And the triplets are going to be the code for each particular amino acid. The example here is GGC, guanine, guanine, cytosine, codes for proline. GCC, guanine, cytosine, cytosine, codes for arginine. So the order of the bases within the triplet determine the code for the triplet. So if you have a triplet code of, if you have a triplet of nucleotides, you have the triplet of the bases. You have 3 nucleotides, one with guanine, another one with guanine, and another one with cytosine in that location on the DNA strand. That location, that triplet code will code for proline. Is the one right next to it, if the triplet next to it has guanine, cytosine, and cytosine as its nucleotide bases, it's arginine. The triplets specify the amino acids. The triplets are made up of the bases, and the bases, the order of the bases determines what the triplet's going to say. So the triplets determine the amino acid and the order the amino acids have to show up in. We don't randomly have a collection of bases. The bases are put in the bases appear in the DNA in a very specific order. Because that's all the DNA is. The DNA is this 6-foot-long strand of nucleotides with their bases. 6 feet of adenine to thymine, and thymine to adenine, and guanine to cytosine, and cytosine to guanine, running as a double helix, for 6 feet, paired up. And on one side of the strand or the other side of the strand, we have these bases that are going to pair up, and they're organized in clusters of 3. And each cluster of 3 is a triplet, and each triplet specifies an amino acid. So when we're talking about a gene being the physical location of the instructions on how to make a protein, that's what we have. If our gene starts with the triplet GGC, it's starting, that amino acid is starting with that protein is starting with the amino acid proline. If the next one in line is GCC, the number 2 amino acid is arginine, and so on. Whatever the order of the amino acids in the protein, that order is specified by the triplets, by the order of the bases in here. So that's what DNA is. The DNA has all the information in the genes. The genes are nothing more than the physical location of the instructions on how to make the protein. And how to make the protein is the order of the bases. Those bases are clustered in groups of 3, and each cluster, each triplet, will specify a unique amino acid.
DNA and RNA are nucleic acids. Kinases made of cellulose called nucleotides. One difference between DNA and
Okay, so each side of the DNA strand, the DNA is a double strand. You can have a gene on one side or a gene on the other side. It doesn't matter. They're going to appear on either side of the strand, and they're going to be in multiple locations. We don't just have one spot only in all of our DNA to make hemoglobin or to make hair color or eye color or skin color or to make a specific enzyme. We have multiple locations. We are redundant. So, but it's the physical the physical location is a gene, and the gene is made up of all these bases from the nucleotide. This is all these represent the bases from the nucleotides in here, and the order of the bases dictates the order of the amino acids that go into the protein. It tells you, like this triplet here tells you one amino acid. The next triplet tells you a different amino acid. The next triplet may tell you the same amino acid. But this process continues from start to finish on each gene. Some genes may have very long orders for a protein with lots of amino acids like hemoglobin, and some may be much smaller. But the order of the bases is very, very important. The order of the bases determines the order of the amino acids in the protein. A mistake in the base will either alter the makeup of the protein, and let me back that up. And an error in the sequence of the bases will alter the triplet. The triplet if the triplet's different, it may change the amino acid. Then a couple things can happen here. It may not be noticed. It may be the code for the same amino acid, which is fine. It may change the amino acid, which may create what we call a junk protein that doesn't do anything, or it may create a situation that's very dangerous. So there's a couple of possibilities here of what can happen. Most of what can happen is not good. So anyway, so the triplet is a triplet is three bases together, specifying a particular amino acid. The triplets specify the amino acids. The order of the amino acids is unique to each protein, and so the bases dictate the triplets, and the triplets dictate the order of the amino acids. So the DNA contains all of these instructions, which amino acid to put where to make a unique protein. RNA has a different role. RNA has to take the instructions from the DNA out to the ribosome. So the DNA never leaves the nucleus. The DNA never goes outside the nucleus, because if it did, it could get altered. We don't want that. We want the DNA to stay constant. So we have our DNA staying in the nucleus, and we simply copy the instructions off of the DNA. That's what the RNA does. The RNA has three roles. First of all, it will copy the instructions off the DNA. The second role that RNA does is to make the ribosome. And the third thing it does is to bring the unique amino acid specified on the DNA, and it will bring each unique amino acid to the ribosome. So we use three different types of RNA. Now they're different a little bit from DNA in the sense that RNA has an extra oxygen in its sugar. That's why we call it ribose, 5-carbon sugar. We call DNA deoxyribose, 5-carbon sugar missing an oxygen. So the three types of RNA are messenger RNA, ribosomal RNA, and transfer RNA. These are the three different types. Messenger brings the instructions out to the ribosome. Ribosomal RNA, as you might have guessed, makes or assembles the ribosome, and transfer RNA brings a specific amino acid to the ribosome when we're making the protein. So messenger RNA, single strand, all RNA is single-stranded. The messenger RNA copies the instructions off of the DNA. The term here is transcription. Transcription means to write down. So essentially, the messenger RNA writes down the instructions in the triplet order. You know, if the instructions are to have, if the DNA specifies adenine, 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 then the complement to that is thymine, 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 and that's what shows up in the messenger RNA. And so the messenger RNA will make a copy of the DNA. It'll be a complementary copy, but it'll make a copy of the DNA in the specific order. And this process is known as transcription. And then ribosomal RNA, as I said, is going to create the structure of the ribosomes. The transfer RNA brings the amino acids in the order that they're specified to the ribosome. They have a triplet code on the transfer RNA that is the reverse of the code on the messenger RNA. Messenger RNA has a triplet. The transfer RNA has a triplet, but that is the anti-code or anti-codon to the triplet. Now that I've got you confused, we'll go on, I'll explain all that. Okay. Protein synthesis takes place in a two-step process. The first step is transcription. This is pretty straightforward. Transcription takes place in the nucleus. The order of the complementary bases, the order of the bases are copied onto a strand of messenger RNA. It's a complementary copy. So if the DNA says thymine, the complement on the messenger RNA is going to be adenine. If the DNA says cytosine, the complement on the RNA is going to be guanine. If the DNA says adenine, the complement is going to be uracil, because we don't use thymine in RNA. I don't know why, but we don't use thymine, we use uracil. So adenine will pair to uracil. Thymine still pairs to adenine because thymine's in the DNA, and adenine will be in the RNA. But adenine pairs to uracil when we're talking RNA. This process of copying the DNA is called transcription. You transcribe the information. When you copy something down, that's a transcription. If you get a recipe from your grandma for biscuits, and grandma doesn't let her cookbook out of her house, she's very reluctant to let her cookbook go. So what you have to do is you have to copy her recipe. You can't take her recipe out of the house. You can't take her cookbook out of the house, but she'll let you copy. So you write it down, or she writes it down for you. That's a transcription of it. It's not the original, it's a copy. It's a word-for-word copy. And that's what we get with messenger RNA. Messenger RNA copies the DNA strand. It makes a complementary copy of the DNA strand. Now, the RNA that we copy is long and has a bunch of what we call non-coding regions in it. Okay, so we have to do a little slicing and dicing to get down to the messenger RNA that we want to use in the ribosome. The actual assembly of the protein is called translation. We are translating the instructions to make the protein. It's like, again, like using a cookbook. The recipe isn't going to make a biscuit for you. You have to translate the recipe. You know, in this case, the translation involves using flour and sugar and salt and water and whatever else, shortening, and to make the biscuit and bake it at a certain temperature in a certain kind of container. And that's great. That's how you make a biscuit. Okay. But if you have the recipe, you're not going to have a biscuit unless you translate the instructions from the recipe into the final product. And that's what translation is all about. It's the instructions on how to make a protein. We translate a recipe and how to make a biscuit. Our cells translate the messenger RNA into how to assemble a protein. So, transcription. You make a complementary copy of one side of the DNA. The DNA splits open. You know, it separates into halves, only at the location of the gene. So you have this double helix. It unwinds, it opens up, and exposes the gene. Remember, the gene is just the physical location of all the bases in the right order to tell you how to make, you know, a protein. So when we're going to transcribe a gene, all we have to do is, it's, you know, pretty amazing how this works. But the DNA opens up, and then we make a complementary copy of the side of the DNA that is the gene. Now, the other side of the DNA can be a gene also for something else. There's lots of information packed into each DNA molecule. I mean, you know, it's a 6-foot long strand of information. And if you purchased a textbook, an A&P textbook anywhere, and you know the A&P textbooks are usually pretty thick, you know, between 2 and 3 inches thick. Imagine if you had those stacked up 6 feet tall. That's how much information is contained in one of those 6-foot long strands of DNA. A tremendous amount of information in here, because we're talking these little microscopic subunits here, the nucleotides. So how do we transcribe? Well, we have to, first we have to pull the DNA off of the histone. The DNA and the histone have to separate. So we have the DNA strung out by itself. So that exposes the gene. Then we have to find the starting point. There is a segment on the DNA called the promoter that tells us where to start. And we find that on, you know, there's always a promoter on the gene. Tell you this is the starting point. Start here. Don't start 2 above or 3 below. Start right here. So now we open up the DNA. We separate the DNA from the histone. We open it up, and now we find the promoters, the starting point, the promoter, and an enzyme called RNA polymerase comes along. We make this enzyme in the cell. So RNA polymerase, a polymer maker, that's what, you know, it's an enzyme, polymerase, is going to start assembling RNA using the original DNA strand as its model. It'll be a complementary strand. So transcription starts out with initiation. The RNA strand opens up. The strand is assembled thanks to the RNA polymerase, and it elongates. It stretches out. It's to the length of the gene. And then it hits a stop signal, a termination signal. So you watch the animation, you can see what's happening here. The DNA has opened up. the RNA polymerase is assembling complementary nucleotides. The red strand is the messenger RNA. You'll notice that adenine pairs to thymine from the DNA, and uracil pairs to adenine in the DNA. So we open up our strand. That's the initiation. We open up our strand, exposing the gene. The RNA polymerase starts assembling the genes. The RNA polymerase starts at the promoter and starts assembling the information, making a complementary copy of the gene, putting complementary bases in order. And it will continue till we get to the termination point. And whether it's 360 amino acids in length or 60 amino acids in length, we will have all the bases necessary for each one of those amino acids. Now, the RNA that's made contains a lot of information that is coding information and non-coding information. We have to remove the non-coding material. Now, we don't know what that non-coding material is there for. We used to call it junk DNA, but it's not. We don't know what it's there for. It's called non-coding. The non-coding material that you see here is that yellow section, and it's not really yellow, but it's called an intron. An intron is a segment of information that is non-coding. So the original messenger RNA has copied everything in the gene. Part of that material that got copied is not going to be used. We don't know why it doesn't get used, but it doesn't. And that non-coding region is called an intron. What we want to have left are the exons, the sections here in green. So what we do is we slice out the introns and we glue the exons together. They use an enzyme, a protein called spliceosomes. If that wasn't a Friday afternoon name, you know, when these geneticists or genetic scientists are sitting around, what are we going to call this thing? Well, it splices together. It's, well, if we call it spliceosome, we can get out of here and go hit happy hour. That's, you know, this image of them saying it's a, you know, instead of some, you know, sophisticated technical name, they call it spliceosomes, but it works. And it's something you don't forget the name of it either, where it splices the two sections of exons together and you've taken out the material that is not codable or not coding. We don't know why it's not coding. It may have another purpose somewhere else. You know, we've gone from junk 10 years ago to non-coding as we know it. So 10 more years, who knows what we're going to find out about the non-coding regions. Anyway, we take out the area that is non-coding is removed. The coding regions are joined together by spliceosomes. And now we have the processed messenger RNA that is ready to go. And it's nothing that we have to do. We have no say in this. When we're getting ready to make a protein, this is how the cell takes out the non-coding material. So, and we'll always do this. What happens to it? Well, we don't know. We don't know what happens. We don't know why we have the non-coding sections in there. For all we know, they are coding something else, and we don't know that yet. But we do know that there is a slicing and dicing effect here. And that's where we get the term spliceosomes. So we've got our messenger RNA now that has been cut down to just the coding regions, and it will leave the nucleus through the nuclear pore and go straight into the rough endoplasmic reticulum, the rough ER. And that's good. That's where we want it to go. And now when it goes to a ribosome, we now start the process of translation. We have to translate the information that's found in the messenger RNA. It's like if grandma makes a copy of her recipe for biscuits, she copies it down word for word, but she leaves out the part that doesn't apply anymore because you're not using a wood stove. So maybe the instructions halfway down specified how to build a fire in a wood stove to maintain 350 degrees. And you have to do this, 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 and this, and that's not relevant anymore. So she leaves that out of the instructions. Well, she's taken that part out. She has been the spliceosome for the instructions that aren't relevant. Or perhaps grandma doesn't want you to have her best recipe and she leaves out a few instructions. Either way, part of the information that has been removed. Is it essential information? Well, apparently not, as far as we know, but it has been removed. So what we get going to the ribosome is the trimmed down spliceosome treated messenger RNA, which has only the information on how to make a protein. All that's in the messenger RNA are the exons that are glued together. The exons have been glued together, and you have the unique information there to start assembling amino acids in their very specific order. The order that the amino acids in a protein is absolutely essential. It has to be correct because little mistakes mean things. We don't like little mistakes. But we get our messenger RNA going out into the ribosome, and now we're going to make a protein. The messenger RNA represents the complementary copy of the bases on the DNA. We still need to have we have to have ribosomal RNA to make the ribosomes. We have to have transfer RNA to bring the amino acids to the ribosomes. On the original DNA strand, we have triplets, clusters of 3 nucleotide bases. It can be adenine, 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 or guanine, 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 or cytosine, guanine, cytosine, or adenine, thymine, adenine, whatever the sequence is. It's not random. It's already been specified. Each triplet sequence is important. Each triplet sequence in a working section of DNA where in the coding sections of the DNA, each working sequence there is unique for a individual amino acid. And so when we make a complementary copy of the DNA onto the messenger RNA, we're giving us the complement in the triplet code. So if the triplet code in the DNA is adenine, 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 the complement on the messenger RNA is going to be uracil, uracil, uracil. That's the complement. The sequence is very important. So you have the DNA strand triplet is reproduced on the messenger RNA side as the DNA's complement. We call the complement on the messenger RNA a codon. Triplet on the DNA, codon on the RNA. So if we say that if the triplet on the DNA is adenine, 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 the codon on the messenger RNA is uracil, 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 because RNA, uracil is complementary to adenine. Now, and you're thinking, well, wait a minute, if I haven't lost you at this point, and this is challenging material. This is some of the most challenging material you're going to hear. Didn't we just change the order? If we had 3 adenines on the DNA, and we have now we have 3 uracils on the messenger RNA. Yes, it's the complement, but it isn't that different. So we'll just be patient. Now, 64 possible combinations. That's what we have. What am I saying here? 64 possible codons. 64 possible codons right now. How did I get 64? Well, if you consider that there are 3 bases per codon, there are 3 bases for codons, and there are 4 bases to begin with. So if you have 3 bases per codon and 4 bases to begin with, that gives you the mathematical progression there gives you 64 possible combinations. You can have adenine, 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 thymine, adenine, adenine, guanine, adenine, adenine, cytosine, thymine, guanine, yeah. So you have adenine, you have adenine, 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 thymine, adenine, adenine, guanine, or adenine, adenine, cytosine. There's 4 different combinations right there, just changing the last nucleotide in that group. Well, if you start replacing, moving them, or if you change the sequence on all 3 positions, just take my word for it. There are 64 possible combinations of codons or triplets. 64 possible combinations. Now there are 3 what we call stop codons that tell the RNA stop making protein. So we have 3 unique stop codons that aren't coding for an amino acid, they're coding for stop. Now what makes it interesting is that there are only 20 amino acids. So if we have 61 codons, that means that we have some redundancy. That means that some DNA strand, some codons are gonna, we're gonna have different codons translating to the same amino acid. You can have multiple codons for the same amino acid. This redundancy allows us to have errors in the order of the sequence and not have it matter. So what do you have to remember from this? There are 64 possible codons. There are only 20 amino acids, so we have redundancy. Now this chart at first glance looks very confusing. I'll be the first to admit that. But what this does is shows you all 64 combinations and the amino acids they specify to. So we have 3 letters in the codon. First letter, second letter, third letter. First letter over here, the left-hand side, these boxes running down the left-hand side. It says U, C, A, and G. Uracil, cytosine, adenine, and guanine. That represents the first letter. There are 4 possible first letters. There are 4 possible second letters. We see here uracil, cytosine, adenine, and guanine. And here are all the possible combinations of the third letter. So, for example, if we are looking up AGA, adenine, guanine, adenine, The first letter is adenine, so it's this row. The second letter is G, so this is this column. So look at the intersection of adenine and guanine. You have 4 different combinations: AGU and AGC, AGA and AGG. Well, it turns out that the first 2 combinations code for the amino acid serine, and the last 2 code for arginine. So it doesn't matter, what the combination is. It usually it's going, whatever the combination is, it's going to code for something. GGC. I'm just making these up right now. GGC. There's the first row is G there. The second row is G. The second letter is G, so it comes down here. And the third letter is C. GGC codes for glycine, but so do GGU, GGA, and GGG. All code for glycine. So we see this redundancy here in that multiple codons can code for the same amino acid. Look at this. Leucine has 6 different possible combinations: UUA, UUG, and then all the C's here. Proline has 4. Threonine, alanine each have 4. Valine has 4. Glutamic acid has 2. Arginine has 6, 4 here and 2 here. So there's also, all these combinations that can occur. We have the stop codons labeled in yellow. They all start with U, uracil. And so you have 3 possible combinations here. UGA is probably the most well-known. If you think UGA, if you're a Georgia fan and you know who UGA is, or you see UGA and you think University of Georgia, well, that's fine. That's the stop codon. There's always a start codon too, over here. AUG in orange. Adenine, uracil, and guanine. It codes for methionine. It's the only one that codes for methionine. That is always the start codon on any protein. So the sequence of the bases on the DNA strand is copied onto the messenger RNA. It's a complementary copy. So whatever base is on the DNA strand at the gene is copied onto the RNA strand as its complement. And the complement, then the codon in the RNA strand specifies the amino acid. Okay. Now, transfer RNA here brings the amino acid to the ribosome. Transfer RNA. Each transfer RNA is a sequence of bases. And each transfer RNA will bring a unique amino acid to the messenger RNA in the ribosome. It will for each transfer RNA, a specific amino acid attaches to one end of it. It's the proximal end, if you will. It's the head of the molecule, whatever. It's at the one end of it. We call it the stem. Okay. So there is the transfer RNA. Now at the head end of the transfer RNA is called the anticodon. The anticodon. So here we see the anticodon. Anticodons will bind to their complementary codon. Transfer RNA, at the head end of each transfer RNA molecule, there is going to be an anticodon. The anticodon will only bind to its complementary codon. So UAU would bind to AUA. Because the complement to uracil is adenine, the complement to adenine is uracil, and the complement to uracil is adenine. So UAU only binds to AUA. These transfer RNA molecules will come looking for their anticodons will come looking for their complementary codon on the messenger RNA that has entered into the ribosome. And they're going to be dragging along their specific amino acid. Now that amino acid is going to be specific for the anticodon, which is the complement of the codon. That's what I just said, the first bullet. The anticodon of the transfer RNA will bind only to the codon on the messenger RNA that is its complement. If the codon is adenine, uracil, adenine, transfer RNA containing uracil, adenine, uracil is the only thing we can bind to it. And so within the ribosome, the transfer RNA will bring in the amino acid. The anticodon on the transfer RNA will bind with the codon of the messenger RNA and bring in that specific amino acid just for that anticodon. Now, the process of translation is very similar to the process of transcription in that you have initiation, elongation, and termination. 3 phases, just like you had in transcription. There is always going to be an initiator. There's always going to be an initiator site at the beginning of the messenger RNA. Always. And so to start the process, transfer RNA carrying methionine will always land on the initiator. Methionine has UAC and it will land on AUG. So if the methionine, if the messenger RNA, if the initiator codon is adenine, uracil, guanine, the transfer RNA that will land on it is the complement. Complement to adenine is uracil, complement to uracil is adenine, complement to guanine is cytosine. So the transfer RNA with the complementary anticodon will land on the messenger RNA on top of the complementary codon. And so this starts the process. This is every protein starts with methionine. It only has one possible combination for methionine. And it will land on the initiator. This starts the process moving. So we always have the transfer RNA bringing in methionine. It will land on its complement, on the messenger RNA in the ribosome. So here we have messenger RNA, and there's the complement right there for methionine. Methionine has the anticodon, and it is the complement to the codon on the messenger RNA. And so the methionine binds to the codon, and a second transfer RNA comes in and binds to the second codon. Its anticodon is the complement to the codon. Then a third one will come in, and it's all specified by the order of these triplets on the messenger RNA. So we start the process by assembling the ribosome. The ribosome has a small subunit and a large subunit. We start out with the initiation. So we have the initiation going, and now we have the remaining sequence of amino acids. The base on the bases on the anticodon are complementary to the bases on the codon, and we bring in amino acid number 2 and amino acid number 3, and we continue on and on until we hit a stop codon. So we have initiation, then we have elongation, and then we have termination and stop. So this is initiation. We always start with methionine. AUG is the codon, UAC is the anticodon for methionine. Here comes arginine. Once arginine, the methionine and the arginine will bind together with a peptide bond, and then it'll kick out the transfer RNA, which will go and get ready to reload and start up somewhere else. Arginine stays there, the arginine and methionine stays there until the third amino acid comes in. Arginine binds to the third amino acid. Now you have 3 amino acids in order, held together with peptide bonds, and it'll kick out the transfer RNA that brought the arginine in. There's methionine on its transfer RNA. Here comes the second amino acid. It binds on the second spot. The methionine and arginine have a peptide bond forming. When that occurs, the transfer RNA is removed. Here comes the third one. This is glycine. Arginine binds to glycine, and methionine and arginine together. They're bound to the glycine. Arginine's transfer RNA is kicked out. Now you have 3 amino acids linked together as we continue chugging down along the messenger RNA. Elongation continues. Transfer RNAs continue to bring amino acids. Transfer RNAs with their anticodons are bringing specific amino acids, and the anticodons will bind to the codons on the messenger RNA. Down here in the red band is the messenger RNA, and the transfer RNAs come in and land on top of their complement, and the amino acids that are adjacent to each other, like arginine and valine, will have a bond, and then the next one comes in here and lands at this spot, and then the valine, arginine, and whatever else was over here will bond to this one, and we'll start building our strand. We'll start building our strand as we go along. Once we hit one of the stop codons, like UGA, like UGA, that stops the process. It stops the codon. It breaks the chain at that point. So we have an initiation codon and a stop codon, and the chain of protein, the polypeptide chain, the protein, is released. So we've used the messenger RNA and the transfer RNA and the ribosomal RNA to assemble a chain of amino acids in the order specified to give us our final protein. And this is here comes, this, the end result is here's our ribosome. Our polypeptide chain grows. When it's done, it is captured in a transport vesicle and then taken to the Golgi. Let me Important point here. The DNA triplets, the base triplets in the DNA are copied onto the messenger RNA as complements. So if the DNA has an adenine, the complement is going to be uracil on the messenger RNA. If the DNA has a thymine, the complement will be an adenine. So we have the codons are the complement of the sequence in the DNA. The anticodons that are bringing the amino acids to the messenger RNA. Remember, the anticodons are the complement to the codons. The messenger RNA has their bases in complementary sequence. The sequence is the complement to the DNA. You know, guanine and DNA, cytosine and uracil. I'm sorry, guanine and DNA, cytosine in RNA. Cytosine in DNA, guanine in RNA. It's the complement to the DNA. The messenger RNA is the complement to the DNA. The transfer RNA is the complement to the messenger RNA, which turns out to be the exact same sequence of the original DNA strand. The anticodons on the transfer RNA is identical to the DNA sequence. So if the DNA sequence was cytosine, 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 and the messenger RNA was guanine, guanine, guanine on the codon. So if the triplet was CCC and the codon was GGG, the anticodon on the transfer RNA is going to be CCC again. So the anticodon brings us back to the original sequence in the DNA. So we really are specifying the order of the amino acids in the DNA. We have to make a complementary copy of the DNA, place that in the messenger RNA, and then we have to have a complementary copy of the messenger RNA to bring us back to the DNA, which is just amazing how this works. So the DNA really is the order, because we're bringing the amino acids to the messenger RNA using the anticodon of the transfer RNA as the complement to the messenger RNA. And the complement to the messenger RNA goes back to the original DNA sequence. So, now, problems with DNA. We talked about mutations. All sorts of things can cause mutations. We talked about, smoking and chemicals and pesticides and pollutants and viruses. All of these can cause problems. Radiation. A mutation is any type of change in the order of the bases on a DNA strand. Here we have a sequence, a gene sequence, cytosine, guanine, adenine, adenine, cytosine, 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 guanine, adenine. That's the original sequence on the DNA. That's the information at on that particular gene. If we put in a different base in here, that's a substitution. So here we see, cytosine, guanine, adenine is substituted for, we substitute, let's see, CCA, we put a cytosine in here instead of a guanine. A substitution mutation. This is not a copy down here. This is the wrong base being put in. The first codon, the first triplet on the DNA should be cytosine, guanine, adenine. But the first triplet that gets showing up here in the DNA is now cytosine adenine. We've replaced the guanine with a cytosine. So we have initially CGA and we end up with CCA. Now, is this going to make a difference? Let me go back and take a look at this little chart here. Oops. So CCA and CGA, CCA. CCA, CC. Makes no difference. CCA and CCA is for proline. CGA, CGA is arginine. It does make a difference. If we put proline, if we take out the guanine and put in cytosine, CGA, which is the original, codes for arginine. It codes for where did I see the proline? Yeah, CGA codes for arginine. The substitution codes for proline. That may be significant. You know, all I'm saying though is that when we substituting one amino acid here, substituting one base causes the change of an amino acid from arginine to proline. Arginine to proline. We see, I'll show you here in a second, that changing in sickle cell disease, the reason the error that gives us valine at the number 6 spot instead of glutamic acid is also a one base substitution. So substitutions may not be important. A lot of times they aren't. A lot of times the substitution gives you the same amino acid. You know, some of those amino acids have 6 different combinations of triplets, and some only have 1 or 2. And so a mistake where a substitution error frequently doesn't cause any harm, but occasionally it does. So a substitution is simply putting in the incorrect base instead of the correct one. Here we're putting in cytosine in lieu of guanine. Okay. Sickle cell disease. Sickle cell disease is a substitution mutation. Here we see it. Normal hemoglobin has glutamic acid in the number 6 spot. It's about 360 amino acids long. We make the mistake. The error is made at the number 6 spot. Doesn't matter that everything else is fine. One mistake early on. It could be late, doesn't matter. It doesn't matter if it's early or late, but it happens. Now, glutamic acid, which has the sequence CTC, GAG, those are its, there's its triplet here, and its complement over here is for glutamic acid. Valine is very similar. Valine is very similar to glutamic acid. You can see that the mistake is simply one base. Here we have adenine. We have adenine here. Here we have uracil. So instead of going cytosine, thymine, and the complement is guanine, adenine, guanine. Down here we have cytosine, adenine, cytosine, guanine, uracil, guanine on the RNA. One base alone changes everything and becomes very important. So substitution errors can be insignificant, but in some cases can be very, very significant in here. Frame shift mutations are even worse. Substitution mutations can happen all the time. And because there's so much redundancy in the coding for the amino acids, many of the amino acids have 6 different combinations. Many have 4 different combinations of codons. So even if there is a substitution, there's a pretty good chance that it's going to substitute to the same acid, or to with 6 different possible combinations. You may, even though you changed your C to a G, it may still work. So occasionally when it doesn't, when there is a substitution, though, you get a significant error. Frame shift mutations, though, are different. A frame shift mutation occurs where something is taken out of the DNA strand, and then the DNA strand slides up one to replace what's missing. So here we see in the top strand, we have the original strand has a codon, a triplet of GCT, AGT, ATA, CGG, and CGA. What happens if we take out one of the a set of bases, or we add a set of bases? Frame shift mutations can go either way. We can either insert extra bases or take bases out. If we've inserted here, right after right after the second triplet, we insert a guanine and a cytosine. The third triplet is supposed to be ATA and TAT. Third triplet is supposed to be ATA. Now we've made it GAT because we put a guanine in here and a cytosine here. So we've gone, instead of ATA and thymine, adenine, thymine, we now have guanine, adenine, thymine, and cytosine, thymine, adenine. So we've totally changed the triplet. And that changes the triplet for everything downstream. Everything has changed by inserting one extra set of nucleotides. If we took out one, if we took out the A and the T, and now the third triplet would go TAC and ATG. Imagine that if we took out the A and the T on the third triplet, A from the top triplet, T from the bottom, we took out A and T, we'd have totally new combinations all the way downstream. And so frame shift mutations are very dangerous. They usually end up stopping the production of the protein. The protein is usually destroyed. It's not, so we're not making the protein anymore. The odds are it's either going to make a defective protein that becomes dangerous, or we have destroyed the gene. We don't like to insert or delete additional nucleotide bases, because from the point of insertion or deletion, everything downstream has shifted. That's why we call it a frame shift mutation. We've either shifted down or we've shifted up. And by shifting, everything above or everything below the shift is now changed, which is not a good thing. So fortunately, it doesn't happen very often. Or if it does happen, remember we have many locations of for our genes. We don't leave all the genes. We don't have just one copy of every gene. We have many copies. So even if a gene is damaged and can't make the protein anymore, we have other copies. We have other copies in other locations that hopefully they haven't been damaged. So that's why the frameshift mutation is so dangerous to us. Because what if it destroys all the gene locations? So, okay, what about cell growth? I've said several times that the DNA in every cell is exactly the same. There is no difference in it. Only the DNA segments that are turned on for that unique cell. So a living skin cell and a kidney cell and a liver cell all have the same DNA, but they're all doing what they're supposed to be doing. They've already been differentiated, developed into a skin cell doing skin cell things, or a liver cell doing liver cell things, or a kidney cell doing kidney cell things. The cells differentiate very early on. When development starts at the point of conception, we have very rapid cell growth occurring, so that we go from one single cell on day 1 of conception to day 7, the date that we implant in the uterus. We've gone from one cell to about 160 cells that are organized into a specific shape and are, developmentally, they've already been programmed to go do what they're going to do. From a differential perspective, they are starting to form our arms and legs and fingers and toes and the head and our skeleton. 6 weeks after conception, we have a complete skeleton in place. 3 weeks after conception, we have a heart that's beating. So we go through a process known as differentiation and determination. Determination means the cell is predetermined to go become a skin cell or a muscle cell. The DNA in that location, and the cells in a specific location are predetermined to go become skin cells or muscle cells or nerve cells or whatever. Cell division, though, can occur at rapid rates or at slower rates. We will have what we call hyperplasia, when the cell numbers grow rapidly at the site of an injury, for example. We always have to replace cells that wear out. We have hyperplasia of white cells when we're fighting an infection as just rapid cell growth, and then we come back down to normal. The other side is atrophy. If we don't use our structures, they're going to get weaker. They're going to get weaker, and they may become more fragile. They don't die off. They just become weaker. They shrink in size too. And cells age. We don't know why cells age. There are a variety of theories here. Wear and tear. Mitochondria are damaged by free radicals. Autoimmune problems. But the one that seems to have the greatest promise is that discussion about the telomeres, the caps on the shoestring structures on the ends of the chromosomes. The telomeres shorten every time the cell goes through mitosis and cell division. And so the cells gradually wear out as the telomeres shorten. They don't grow back. Cancer cells can keep their telomeres growing. Cancer cells are hard to kill. We can kill them, but it's hard to kill them. And the cancer cells have an enzyme called telomerase, and that's what keeps rebuilding the ends of their chromosomes. Healthy cells don't have the telomerase in here. So that's to me. This is my favorite theory on cell aging. the increase in the shrinkage of the telomere in healthy cells. And let's see, okay, that is as far as we're going to stop because we're done. And I hope I haven't been too boring on this. So on Tuesday, we're going to start chapter 24. Chapter 24. It's in the old textbook, it was 24. It's a chapter on metabolism, and it's going to go fairly quickly. And then once we get done with that, we're going to start moving into the organ structures. The first thing we're going to look at is the physiology of bones. How do bones work? And then we're going to go into muscles. How do muscles work? So anyway, and that is everything I had to say. Are there any questions? Okay, I want to get us out of here, and I will see you all later. Have a great weekend.